Hello, my name's Hadden Graham and I'm the Global Technical Director of AB Vista. I would like to talk to you about one of my favourite topics, and that is dietary fibre, and how an understanding of dietary fibre can help you to optimise your feed. So why am I interested in dietary fibre and carbohydrates? Well, if you look at the dry matter of most compound feeds, you will see somewhere around 5% or less ash, around 5% fat, and maybe 20% protein. That leaves 70% unaccounted for, and that 70% is primarily carbohydrates. And those carbohydrates can be split down into 4 categories. The first category is in monosaccharides. In animal feeds, that's primarily glucose, but you do find some fructose. Then you have the disaccharides. Again, in animal feed, that is primarily sucrose. But for example, in piglet feeds, you also find lactose coming in milk products. The oligosaccharides we tend to spend a lot of time talking about, and when we do, we often mean the fructooligosaccharides and the raffinose series, which includes raffinose, verbascose, and stachyose. And then we come to what is generally the major carbohydrate in any feed. That includes the starch, and then the 4 other polysaccharides, which are pectins, fructans, hemicellulose, and cellulose. And those 4 make up what we know as the dietary fibre. I won't spend too much time talking about fructans, because fructans are quite acid labile. That means they are broken down to some extent as soon as they enter the stomach, and they are degraded quite quickly by the bugs in the animal gut. So the fructans function primarily as an oligosaccharide, not as a dietary fibre, so I will not discuss fructans in the rest of this video. If we look at the definition of dietary fibre, there is a classical physiological definition that dietary fibre is the part of the diet which is not broken down by the animal's digestive enzymes. That is a very good definition, a very clear definition, but unfortunately, if you're an analyst, it doesn't allow you to analyse dietary fibre. So what we have then is a chemical definition. The chemical definition says that dietary fibre is the sum of the non-starch polysaccharides plus lignin. And in effect, these 2 definitions come up with quite often the same answer. If we look at the analysis of dietary fibre, we can take the feed, we can add enzymes to it, for example, amylase, protease, lipase, digest away all the nutrients, and weigh the organic matter which is left. And that would be the dietary fibre. We can then split that down further into lignin and non-starch polysaccharides, or we can split it even further into pectins, hemicellulose, cellulose, plus lignin. There are 3 other methods to analyse fibre which are well known and have been used for a number of years. These are the gravimetric methods: crude fibre, acid detergent fibre, and neutral detergent fibre. The crude fibre method is around probably more than 100 years old. It involves boiling the feed in dilute acid and dilute alkali. That recovers the majority of the lignin and the majority of the cellulose. The ADF, or acid detergent fibre method, involves boiling the feed in a dilute acid with a detergent added, and that captures also most of the cellulose and most of the lignin. And then the NDF, or neutral detergent fibre, captures the cellulose, the lignin, and the majority of the hemicellulose. Note, none of these 3 gravimetric methods capture the soluble fibre. So let's look at the carbohydrates you can see in 2 common feedstuffs. Here we're going to look at corn and soybean meal. The primary carbohydrate in most cereals is starch, and here we can see it represents close to 70% of the total grain. If you look at soybean, on the other hand, there is almost no starch and there's very little levels of fructans, but the main component, or the main difference between soybean and corn, is the 5% oligosaccharide you can see in soybean meal, and that is primarily raffinose, stachyose, and verbascose. If we then look at the fibre fractions, we can see in corn that the crude fibre and the ADF are similar. They both capture the cellulose and the lignin, and that is fairly representative of the whole fraction of the corn. NDF is around 9% and represents around 90% of the total NSP plus lignin, which makes up the dietary fibre. So when we see that the NDF is 90% of the total fibre, we can conclude that there is little soluble fibre in this corn sample. If we look at the soybean meal, on the other hand, we see a slightly different picture. We see again that crude fibre and ADF are quite similar, 4 to 5%. We can see that NDF is around 8% and capture is only 40% of the total dietary fibre. And this is because the pectins that you find in soybean meal are soluble in NDF, and therefore the NDF represents a much lower fraction of the total dietary fibre. If we then look at the non-starch polysaccharide residues present, and let's look first at corn, we can see the predominant sugar here is glucose, and this glucose comes from the cellulose. Next then you have the arabinose and xylans from the arabinoxylan, and that is from the hemicellulose. If we look across a soybean meal, we again see that glucose is the primary sugar, and that comes again from cellulose. However, we see some other sugars now coming in, galacturonic acid, galactose, and again arabinose. And in this case, these come from the pectins that you find in soybean meal. So dietary fibre can be both soluble and insoluble. The soluble fraction tends to increase digestive viscosity. In the animal, that can prevent the animal's digestive enzymes moving, it can prevent the nutrients moving, and therefore can reduce digestibility of the diet. It can also cause some welfare problems, such as wet litter. These soluble fibres tend to be totally broken down as they pass through the animal's tract, and therefore you produce volatile fatty acids that can be used by the animal for energy. The insoluble fibre, on the other hand, is very different. It bulks up the digester, it tends to increase passage rate, and it tends to increase, for example, gizzard activity. These insoluble fibres can also encapsulate and prevent the digestibility of nutrients such as starch and of protein. Insoluble fibre is only partly degraded and therefore provides much less volatile fatty acids and much less energy to the animal. Here we have a scanned electron microscope of wheat, and here we can see an example of what it means when I say that the insoluble fibre encapsulates the nutrient. In this SEM, the fibre is stained blue or fluorescent blue, the protein is stained as red or brown, and the starch appears as starch granules as black. And this is a two-dimensional picture, but from this picture you can imagine that the blue fibre surrounds and encapsulates particularly the starch and the protein and prevents the animal's amylases and proteases entering these cells and digesting the nutrients. This is what I mean by encapsulation. If you add an enzyme to this type of diet, what the enzyme can do is break down those insoluble fibres, open up those cell walls, allow the animal's own enzymes to enter and degrade the nutrients such as starch and protein. The fibre degrading enzymes will also quite quickly reduce the viscosity. That will allow the animal's own enzymes to move quickly in the gut. It will allow the nutrients to move quickly and will allow much quicker and better digestion. Both of these actions will produce oligosaccharides. These oligosaccharides will be degraded further by the bacteria in the gut and will provide energy for the animal. This will also encourage the growth of what we call healthy or good bacteria in the gut. Note that I did not say that the fibre degrading enzymes could break down the fibre to sugars. This does not happen in the animal gut. It is extremely difficult to break down fibre to free sugars, particularly within the time limits you face during passage of feed through the animal gut. And any oligosaccharides produced by the enzymes will quickly be degraded by the bacteria present into volatile fatty acids before the enzymes have the chance to break them down further to free sugars. However, this is positive. If we look at the next figure, we can see here from an animal or broiler trial done in Holland back in the 1980s, and here they have looked at adding 5 different levels of 3 different sugars. And the 3 sugars they have chosen here are firstly glucose, that would represent the degradation of cellulose. Then we have xylose, which would represent the degradation of arabinoxylans. And finally, we have arabinose, that will represent the degradation of pectins or arabinoxylans. And we can see the birds grow quite happily when they have glucose added to the diet. However, when you have arabinose or xylose, the bird's growth rate decreases significantly. And that is because the animals also reduce intake and start to drink more water, because these pentoses are very poorly absorbed and metabolized by the animal. So you do not want to break down, for example, pectins or arabinoxylans to the constituent sugars within the animal. That can be negative for animal performance. So to sum up, feed carbohydrates are well defined and they can be accurately analysed. However, the analytical results don't necessarily tell you what those carbohydrates will do in the animal gut, and that is particularly true of fibre. For example, soluble fibre is very different than insoluble fibre. Cellulose is very different from pectins. Fibre can be both a nutrient in that it can be degraded and it will provide energy to the animal, but it's also an anti-nutrient in that it can increase gut viscosity, it can encapsulate nutrients, and therefore can reduce digestibility in the animal. Xylonases tend to be the most effective non-starch polysaccharide enzyme used in animal diets. That's because the arabinoxylans tend to be the main encapsulating fibre in animal feeds, and it also tends to be the main viscous fibre. And finally, complete degradation of fibre to free sugars in the animal gut by feed enzymes is neither possible nor desirable. Thank you.